Thank you, Serena. And now we have Jean-Claude Manuguerra from the Institut Pasteur in Paris. Please, Jean-Claude.
Thank you, Daniel. I want to thank the organizers for inviting me. It's a wonderful place and wonderful company, you know, as well in the room and in the restaurant. So it's very nice. So I'm going to talk to you about the role of existing public health and research laboratory networks in pandemic crisis. And first, I would like to talk about the time factor in pandemic response. We saw and we heard from previous speakers that, you know, the time goes fast, that the speed of event is very fast in a crisis reality. And if you take the last two pandemics to date, the H1N1, swine flu, Mexican flu, I don't know what you want to call it, or flu A, it takes between the identification of a new virus in Mexico mid-April 2009 to the declaration of the pandemic less than two and a half months. If we look at COVID, it was the same, from the first clusters to the declaration end of January of a public health emergency of national concern. It's a bit more than a month, and in March 11, already the pandemic was full-blown. You have to bear in mind that at the time of plague, at the mid of the 14th century, the plague in Europe was progressing from the east at 1.5 kilometers per day, and it took it 48 months to reach Europe. Spanish flu, it took about 4 months, and about the same for COVID for the whole planet. So, you know, they have to take this into account. And then what is crucial is the networking. Because of the geographical spread of the outbreaks, and we can afford to build capacity during a crisis. And pre-existing networks have the advantage they are already operational and actionable. Now I'm going to talk about 3 different networks, Medilab Secure, Durable, and the EU Reference Laboratory for Public Health. I want to start with Medilab Secure, which is a network for promoting One Health approach to mitigate biological risk related to emerging vector-borne viruses in the Med, Black Sea, and Sahel region. So the emerging disease, you heard that since the beginning of this conference, are very important, but you know, we know that some are zoonotic, but a part of them are borne by vectors, and this represents about 25 to 30% of the emerging infectious disease. And this is a challenge because of their complex ecology, and it's linked also to the affected by climate change. The vector-borne disease infects millions of people per year, and account for about 700,000 deaths per year. So in the agent, you have not only, but mainly arboviruses, chikungunya, dengue, Rift Valley fever, I'm going to talk about this later, yellow fever, Japanese encephalitis, West Nile, you name it, Congo hemorrhagic fever. You had also some bacteria, some very deadly, like Yersinia pestis, and you have also some parasites, like malaria, for example. They can be borne by mosquitoes or other insects, like sandflies or fleas, but they can also be borne by ticks. We have disease that affects humans, and they have disease that affects animals, and sometimes they are common. Now, the Medilab Secure had 3 phases. Phase 1 from 2014 to 2018, and the phase 2 and 3 from until 2025. And it's not dead because it's revised like Phoenix in a new project, but it's the same contours. It's called now One Health Secure. But let's see what we've done. So 6 European partners, and this is a project financed by the Center of Excellence, which is an initiative of the European Union. So 6 European partners, experts in their field, collaborate together to promote a One Health approach. And this is a real One Health network. We have human virology led by Institut Pasteur. The whole program is coordinated by Institut Pasteur in Paris. You have animal virology, which is led by the National Institute for Investigation in Animals and Food in Madrid. Medical entomology, which is led by Institute for Research and Development in Montpellier. And we have public health. 2 kinds of public health: human public health and veterinary public health. Human public health is coordinated by the Institut Superiore di Sanità in Rome, and the veterinary side by the Istituto Zooprofilattico de Teramo. And we have also now the spatial modeling and disease management, which is coordinated in Belgium. But the beneficiaries are not these countries. We have 22 beneficiary countries, basically situated in 3 regions. We have the Balkans and the Black Sea, for example, with Georgia, all non-EU countries. You have the Middle East, with Turkey, for example, and we have the North Africa and Sahel region. In total, 111 laboratories, 60 laboratories, and 40 public health and veterinary institutions. They gather about 170 multidisciplinary experts, so 5 regions and 5 networks in each of these countries. How do we work? So we do capacity building mainly through training, and we've trained in the diagnostic of 8 different arboviruses in 10 years, plus 2 coronaviruses. We do training in sequencing, the old one, Sanger, but also the new generation sequencing, Illumina and nanopore technology. We also train in data analysis. We did this for Sanger data and NGS, so new generation sequencing, and also, of course, biosafety and biorisk management. We organized external quality assessment, 16 programs for Zika, dengue, chikungunya, West Nile, MERS, and SARS-CoV-2. We also provide with technical support for preparedness. We had, in 10 years, 18 suspicions of outbreak, 3 epidemics with Zika, MERS, and Mpox, and 1 pandemic, and you know that which one. We also have produced guidelines for One Health implementation and tools. We had produced more than 20 standard operating procedures for diagnostic sequencing. But this led the path to the preparedness to the pandemic preparedness. We went from preparation to response. So COVID-19 is out of the scope of the network. However, that was an emergency, and the European Union has the flexibility to allow us to intervene into this crisis. As soon as January 2020, we communicated and gave information through our network, creating a WhatsApp channel to discuss problems we were facing. In February 2020, we did an evaluation of needs by questionnaire. We shared protocol, and between February and April 2020, we shipped reagents with positive controls, primers, and probe for QRT-PCR. Total 34 shipments to 21 countries, 16 from Medilab Secure, which is 46 kits, allowing 23,000 QR-PCR tests to start with. Now, when we asked the question to our members, were you involved with the identification of the first case of your country? Out of 22 countries, 10 said yes, and we used the reagent sent by you with the protocol. Yes, but we use another protocol. Yes, but we use our own protocol, so this is 5. And basically, nearly all of them participated to the first identification of the case in their country. In the network, 100% of human virology laboratories were involved in diagnostic from SARS-CoV-2. And 60%, and this shows the real One Health side of the network, of animal virology laboratory were involved in diagnostic for human and animals. And this is quite striking. In Spain, for example, the Veterinary Institute did nearly 500,000 diagnostic for humans. Now in summer, we organized 2 EQAs on SARS-CoV-2 detection. And again, we did some training on sequencing the different variants. You know, that was a problem. On public health side, we helped strengthen surveillance and protection capacities with online training and studies on the impact and surveillance. Now, during the same time, we had to face in 2020 an outbreak of Rift Valley fever in Mauritania. And this affects humans and animals, with 75 laboratory-confirmed cases in humans in Mauritania and 138 cases in animals. This disease is caused by an arbovirus that is transmitted to humans through contaminated parts of blood from animals or by mosquitoes of two different kinds of mosquitoes. Now, we had a very straight timeline where we organized a meeting after we were asked by the national health authorities to laboratories, for assistance, and we activated our contacts through meeting over the different networks, that the different colors you can see there. We shared SOP. We contact local beneficiaries of other network to participate to the response, and we ship reagents for QRT, PCR, and ELISA for identification of cases. And later we did the sequencing in Paris, and we had also a visit on site to review local biosafety and diagnostic procedures. So I'm going to go to a different network, and this network is called Durable, for Delivering Unified Research Alliance Biomedical and Public Health Laboratory Against Epidemic. So Durable is much shorter, and this is a HERA laboratory network. I won't detail the mission of HERA because Laurent Michel did it very well yesterday, but I put this slide to remind us in the network that we need to fit the mission of HERA, and we have to have this in our mind. So Durable is 28 partners in 15 EU countries, 200 researchers, and a budget of 30 million, of which 80% is paid by the European Union through HERA. Where do we stand? We stand there in the middle. We do a pathogen characterization. We study immunology. We develop a diagnosis. We also have animals model for transmissibility of the virus. And in modeling, we have a strong component, and we do modeling that is applied. For example, how the PCR results can be predictive of the trend of the epidemic, of the pandemic. If there is a shortage of medical countermeasures, how to use them to have the maximum impact. So this is kind of modeling that we are developing to help HERA in this case of situation if that would occur. So durable interaction with HERA, it's a back and forth relation with HERA, with action and decision, and Durable has to adapt its capacity, its capability to this direction. We have also to produce knowledge and to support decision and fill knowledge gaps. In 2.5 years, we had more than 60 interaction with HERA, some regular, some ad hoc meetings, and we had 6 urgent requests from HERA. For the H5N1 in fur animals in Finland, and also for the Mu flu in the US, and we had to qualify the antigenicity of these viruses and the adaptation or not with the vaccine that has been stockpiled. Now, the challenge of Durable is the fact that we are interacting. So in green, you can see them, it's the situation in Durable, and in brown, each dot is a network, an EU network or a global network. We are in collaboration, in contact, direct or indirect, with 83 networks in the EU and worldwide. So this is something we have to manage and to make it work. So this is the Durable evolution. We try now to fit a bit better with the way that HERA is organized in the 3 categories of groups: respiratory, direct contact, zoonotic, and vector-borne. We develop cross-cutting themes that are listed here. I don't have time to read them all for you. And we have to give contribution to HERA in peacetime doing basic research, but in time of crisis, we want to do outbreak-embedded research. Now, to finish the 38 seconds I have left, I want to touch on the EU reference laboratory that was also touched upon by yesterday. They are new, and they were created after COVID on serious cross-border threats to health. And this is a group. 6 are already operational, but they just started, and they're with 3 coming very soon. One of the most recent ones starting is the one on respiratory viruses. Some of them are composed with institutions within, they are also within durable. And in conclusion, I would say invest in peacetime. Laboratory network require years to mature, so build now to next crisis. Embrace complementarity regionally, continentally, functionally, because these networks create resilient system. Sustained funding, continuous operation maintains technical capacity and trust infrastructure. Integrate One Health, human, animal, environmental surveillance networks to detect emerging threats earlier. Be flexible, like the EU has been with Medilab Secure, and replicate successful models. Sorry, and I'm finished with that.